You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech Podcast, and I have Doug Losordo. He's the Chief Medical Officer at a company called Caladrius Biosciences. Their website is Caladrius.com. It's spelled Caladrius. Doug, thanks for coming.
It's my pleasure. Thank you very much.
Yeah, tell me a bit about Caladrius. Where does the name come from, first of all, and then what's the premise of the company?
Yeah, so the name of the company is actually a very nice reflection of our overall vision. The bird from Roman mythology, the Caladrius bird, is a bird that flies in to a person that's sick, absorbs the sickness, and flies away. It really does reflect very nicely with a nice image the goal of the company, which is not to manage disease, but to actually restore health. That is the vision of the company. That's what our technologies are designed to do, to bring people back to a state of health.
That's a lot better than just treating people in palliative care. So what are the main initiatives of the company right now?
Well, so it's really a very exciting time for the company right now because we're in a nice position of having some late-stage programs that have already demonstrated a very nice efficacy in prior clinical studies, and the focus is cardiovascular disease. As you know, cardiovascular disease is very prevalent around the world, and especially in the United States and other developed countries. And we're targeting the sickest of the sick patients with our technologies. So the idea is to take people who have exhausted all the available therapies, bypass surgery, angioplasty, stenting, medicines, cholesterol lowering, you name it. They've done it all, and they're still disabled. And what we've been able to show in prior studies of our technologies is that a single dose of our therapies does what I alluded to, makes people better long term. We're in a stage now where we're performing a pivotal study in Japan for people with what's called critical limb ischemia. That is limb-threatening ischemia. So the patient has some gangrene or severe pain all the time, which is a harbinger of a future need for an amputation. And that's in a pivotal trial that we expect to complete enrollment this year. And if that study goes the way that prior studies go, we expect that to be an approved product in Japan. So the other pivotal program here in the United States is for patients who have unremitting chest pain called angina. So it's due to a lack of blood supply to the heart. The patient is able to do little or no activity without being stopped by chest pain. And again, they've exhausted everything that's available and are still disabled. And in general, these are middle-aged patients. The average age of patients we've enrolled in our studies are around 62 years old. And prior studies have shown that a single dose of our product results in improved exercise capacity, reductions in chest pain, and actually improved longevity. So the patients live longer.
So starting with limb ischemia, what causes it? It causes it to progress to the point where a limb is amputated, and then let's talk about it. What's the mechanism by which your product helps?
Sure. So this critical limb ischemia is the end stage of peripheral artery disease, which is actually relatively common. By the time people are in their 60s or 70s, around 10 to 15% of people have some disease, we'll just call it blockages, in the arteries that supply their legs with blood. Critical limb ischemia is the extreme of that condition. So they have many blockages such that the blood supply to the leg is extremely limited. And it results in either pain. We've all sat wrong on our leg and the leg falls asleep and then it starts to hurt. Well, that's what those patients feel like all the time. And they need to take narcotics for pain relief just so they can get some sleep at night. Or, as I said, they start to have some gangrene or tissue loss, and that can really, unravel and lead to the need for amputation. Our therapy in both cases is very interesting in that it's a naturally occurring vascular, that is blood vessel, repair cell. So the CD34 cell was shown by research that was done in my lab around 20 years ago to have this natural ability to build new blood vessels in areas where the blood vessels had been lost or damaged. And so, you could almost say this wasn't really so much of an invention. It's not like we invented a drug, but simply made an observation that no one had made before about the ability of these naturally occurring cells to grow new blood vessels. So it's a so-called angiogenic mechanism of action. And interestingly, when we started this work, we thought that we would be doing many studies to figure out proper dosing and so forth. But as we started treating patients, we found that if we gave a sufficient dose, a single administration of the cells targeted into the area where blood flow needed to be improved resulted in very dramatic improvements in symptoms and all the benefits that you would hope for in these different conditions.
Are these cells that, I think they call them autologous, you're removing them from the patient through blood draw, Exactly. You're culturing them and then putting them back in?
Correct. We're not even culturing them. So we concentrate the cells. So we do something to mobilize these cells. The CD34 cells mainly live, if you will, in the bone marrow. That's kind of their home, and that's where they're in the highest concentration. To get a sufficient dose, we give the patients a medicine that mobilizes those cells, perform a procedure. It's called apheresis. It simply collects the white blood cells without removing any red blood cells, collects white blood cells, and then we purify the white blood cells down to the CD34 fraction, which is it's a small percentage, but we're able to get a pretty high number of these cells and concentrating them, put them into a special suspension medium that makes the cells potent, and then deliver them into the target tissue. And for critical limb ischemia, it's really basically an outpatient procedure where the cells are injected into the lower leg of the patient. And then in the refractory chest pain patients, the cells are actually directly injected into the heart muscle, which is where they're needed to build the new blood vessels.
So these cells help to induce angiogenesis and create new pathways, new vessels?
Correct. Yep. So the initial observation in the laboratory suggested that was a capability of these cells. Obviously, we did a large number of preclinical studies before we started any human trials to establish that, in fact, these cells, when delivered into tissues like the heart or the leg that had been robbed of their blood supply, that those cells would stimulate new blood vessel growth, would improve blood flow, and alleviate the condition that we call ischemia, when there's a lack of oxygenated blood.
Well, since these cells are present in the body, it would seem like the body's doing this just not at a significant enough level. I would bet the body is trying to repair any ischemia always. So why is it that the body doesn't do this normally? Why do you have to boost it by concentrating these cells?
Yeah, it's a great question. It's one of those things that, we've observed, as clinicians, we'd make the observation, one patient comes in with some blockages and, maybe you do an angioplasty or a stent and they go home and they're fine. They're out, playing basketball or tennis or whatever. Another patient comes in with what looks like the same exact anatomy of the vessels. You do the same procedure and for some reason they get worse, they deteriorate, they become disabled. So there's something about the difference in the native biology of those patients, their ability to naturally recruit and deploy those cells, that has failed. And we don't yet understand the mechanisms, because of course if we did, right, that would be a better solution, right? Figure out who are the patients who've got a broken CD34 system and figure out the way to get those cells to behave the way they do in patients who are able to repair themselves. We haven't figured that out yet. And so, we're taking the very direct approach of saying, okay, well, somehow these patients aren't mobilizing and recruiting these cells the way they should. So we're going to do it for them while we're still trying to figure out, the mechanisms that have failed that really would allow us, I think, to undertake a preventative strategy. We're not there yet, but I think we will figure out at some point why patients fail to use these cells the way that they were, it looks like pre-installed by the manufacturer. That's what they were supposed to do, but somehow these patients aren't leveraging that biology.
What about the nature of the new vessels created? I don't even know where, I'm just pulling this out of thin air, but I seem to remember reading that when blockages happen or, you get a constriction and new vessels form, that they're of a different nature. They may not be as strong, they may be weaker and more prone to, to rupture.
Yeah.
Or is that the case? Or are you seeing new vessels that are perfect?
I'm impressed. That's, you're going deep into the scientific literature.
Thank you.
That literature that you're citing was mostly done, it is angiogenesis research. It was mainly done around the use of specific gene therapy to stimulate new blood vessel growth. And the implication was that those vessels, like you just said, are more fragile or maybe leaky, and so on and so forth. And those, I would say, parenthetically, were done in some animal models that really, I don't think, had a lot of fidelity for what we're doing. But apart from that, the evidence that we have using these cells is that the vessels that are formed are durable, behave normally, result in improved blood flow. And this is in the preclinical models where you can take the organ out and examine the vessels and so on and so forth. So using the cells to stimulate new blood vessels seems to result in a different type of vessel formation than trying to pick specific genes to stimulate new blood vessel growth. And then I think, really more importantly, obviously from a clinical standpoint, we have 2-year follow-up in a lot of these patients. In some cases, in some of the critical limb ischemia studies that were done, 4-year follow-up. And that to me is the most exciting thing, is that these patients get 1 dose of their own cells deployed into a target tissue, and 2 to 4 years later, they're still better. So the implication there obviously is that whatever blood vessels are formed by a dose of these cells seem to be quite stable and continue. And really, if you look at some of this data that's been generated, especially in the cardiac patients. So if you measure symptoms, chest pain symptoms, and patients treated in these blinded studies, 2 years after a single administration, not only is the chest pain better in the treated patients than controls, but the difference between treated and control subjects seems to be getting greater over time, which sort of suggests that you give a dose of the cells. And we know they take up long-term residence, not a lot of them, but a percentage of the cells that are put in there take up long-term residence and seem to be stimulating an ongoing repair process. And the same thing when we look at mortality. The mortality curves, if you do, say, bypass surgery on somebody, after you get past the postoperative period, you can see that there's a difference in mortality in properly selected patients who get bypassed. But then over time, that difference wanes. When we look at the patients we've enrolled, those mortality curves are diverging at 2 years, so they're getting further apart. So, the implication is that whatever process is triggered by deploying these natural repair cells seems to be long-lasting. We still need to do even longer follow-up. We'd like to do 5-year follow-up on our next round of patients, but so far so good. It seems to be that we're developing very durable functional vessels.
Have you tried injecting sites that are, I guess, quote unquote normal?
Yes.
Does this
That's a great question.
Yeah. Does this?
Yeah.
Yeah, because I can think athletes will be like, oh, inject this into my heart. I want a super heart.
Yeah. So I mean, I don't want to disappoint Lance Armstrong or anything like that, but if you put these cells into normal, healthy well-functioning tissue, they disappear very quickly, because again, we're just leveraging natural biology. So these cells were built to repair blood vessels or damaged tissue that needs a better blood supply. They very quickly can sense if there's ischemia, if there's a lack of oxygenated blood, they migrate toward it and they stick around. If there's no ischemia, and we did these experiments because we wanted to see, how the cells behave. You deploy these cells into normal, well-perfused tissue, they disappear and go right back to the bone marrow.
Well, if you have a patient, let's say that's, 70, they're probably going to have, like you said, some level of ischemia in many parts of their body. I don't know, is it even ethical, or would someone like that be able to request and say, hey, I'd like, 6 or 7 injection sites around my body in small amounts to tune me up and make me young again?
Yeah, no, you couldn't do that, I don't think. I certainly wouldn't do it. And I don't think there would even be a rationale for it. I think that we know these cells will fix ischemic tissue. And as I said, we've really targeted our development efforts so far on the sickest of the sick. So in patients who've got really bad chest pain or heart failure or critical limb ischemia or other forms of ischemic disease where nothing's working, those are the folks that we've concentrated on so far with a very consistent track record. I think that across multiple indications now, in properly selected patients with real ischemia, these cells have yet to have a failure in which they did not show evidence of bioactivity. And of course, safety. We're using the patient's own cells, and so it's almost a given that they're going to be well tolerated. But nevertheless, it's important to point out that the safety track record of these cells is not only unblemished, but that the patients who get the cells experience fewer adverse events, fewer hospitalizations, fewer heart attacks, fewer strokes, the whole thing. So just what you would hope to see when you give a patient a booster dose of cells that are there in the body, it seems, to repair damaged tissue.
Yeah, I wonder if this should be coupled with any stent or with any surgery to, balloon angioplasty or anything, just to make sure that there's no complications from it or problems.
Yeah, look, there is discussion about that because one of the concerns when a bypass procedure is done, especially in the legs, you do a bypass surgery in the leg, and the surgeons will tell you that one of their big concerns for survival of bypass grafts is what they call outflow. I call it the microcirculation. If there's stagnant blood flow in a graft, then we know that graft is at a higher risk for thrombosis. So what you just said, the idea of not only fixing the big blood vessel, but actually repairing the downstream so that there's, repair of those microvessels that have been damaged, improving blood flow through the graft. There's a rationale to think that could lead to better long-term performance of those grafts. It probably is a study that we'll do at some point because the rationale is so solid. It makes perfect sense.
This might be a basic question, but what happens when, let's say you're growing a new vessel? I mean, essentially it dead ends until it connects to the rest of the body, or do these new vessels grow just out into like the interstitial tissue, let's say parallel alongside an original one?
Yeah, I failed to explain this adequately. So the blood vessels that we grow are not the large conduits. They are truly the microcirculation. So the tiny, capillaries that are a big part of all ischemic diseases. When there is blockage of big blood vessels, there is damage to the microcirculation. And, bypassing the big blood vessels certainly is helpful and necessary. But if and this is it goes to the story I was telling before of the 2 patients who have different clinical courses, patients who fail to repair their microcirculation, who have ongoing damage to it, just get worse. And so these cells are like a network of tiny vessels that merge with each other and then merge with downstream vessels that are there. So, we're leveraging a natural biology in which these cells know how to communicate, they crosstalk, they know how to find the tributaries to connect to. And so it's not like, a giant tube that needs to find its way to another tube to hook up to. It's really these tiny vessels that are so important. The analogy I use is, if there were no side streets, nobody would be able to get home at night. If all you had were interstate highways, it would be really hard to get around. And so we're fixing the side streets and the driveways.
Well, I'm imagining in my mind a vessel that's, nearly completely clogged. And the microvasculature, like you said, is like starving, downstream of it because it's not getting enough blood. And the heart probably the whole system's blood pressure probably has to drastically increase in order to force enough blood through there. And then once you put in a stent or you open it up, now all of a sudden it's like this gush of blood going into maybe damaged, microvascular tissue. Maybe that's why some people have a problem, because maybe this sudden gush of blood and this, the pressure maybe hasn't stabilized. Maybe it finishes destroying the microvasculature that was damaged and it needs new stuff to fix. I'm just one step out of thin air, but just something to consider.
No, look, I mean, that what you just described is actually that sort of dramatic example of what you just described does happen. Fortunately, it's relatively rare, but it is one of the things that surgeons worry about, or peripheral interventionists worry about is, if I open up a giant vessel and provide this head of pressure down into the tissue, is it going to cause problems? And occasionally it does. The patients will develop a compartment syndrome, and it can really be a very difficult situation. Most of the time, fortunately, most of the time in those patients who have severe peripheral artery disease, when you're doing a bypass, you're obviously improving blood flow, but not to the dramatic extent that, I think you're imagining that could cause that damage. But it kind of does happen. Fortunately, it's rare.
Well, it seems like, your solution should be maybe preemptive, or before, let's say someone has a blockage, if they don't need to immediately have it addressed. What if they were to use your protocol beforehand to grow some new microvasculature, so when it's opened up, there's not such a dramatic change now, and, like you said, the pressure and the volume of blood flowing.
Yeah, no, look, I mean, I think when I've started to talk to vascular interventionists and surgeons about this is exactly what we're talking. A dose of cells first, wait a few months to let those cells start to work their magic on the microcirculation, and then one of two things will happen, right? Either the patient's completely better, right? Pain's gone, ulcers are healed, gangrene's gone, in which case you stand down. Or if there's improvement but not complete recovery, then go ahead with the bypass operation. But yeah, you've kind of set the stage for a more successful operation. I think, that's exactly the lines that we're thinking along.
And you guys have gone to Japan because from what I've heard, they have a fast track program to approval for certain kind of medical interventions like this, right?
Yeah, that's right. So Japan, in the wake of the Nobel Prize going to Yamanaka for the discovery of the iPS cell, or not discovery of it, but, inventing the iPS cell, if you will, Japan became very interested in identifying itself as a regenerative medicine epicenter for the world. And so they changed the regulatory rules for the approval of regenerative medicine products. Our CD34 cell is clearly regenerative medicine. And then on top of that, I would say that, during my academic career, a lot of the postdoctoral fellows that I worked with were from Japan, just by coincidence. And in fact, the postdoc who really made the seminal discovery about the CD34 cell was also from Japan. And so there was actually a lot of momentum in Japan to perform this development program. Some studies on CLI had already been done, and so there were a lot of reasons why Japan made sense. Now, interestingly, of course, since that time, the U.S. and Europe have tried to create their own systems for accelerated approval of regenerative medicine. So in the United States, the 21st Century Cures Act and the whole RMAT designation were created. Our program for refractory angina has been designated with the RMAT designation. And so that's one of the tools that we're now leveraging here in the United States. But yeah, the reason to go in Japan was because they were ahead of the rest of the world by about 4 years.
That's great. Are there any? I don't remember the name of the cell. I think you said it was CD34 or CD38 cell. What was the name?
CD34. Yep, 34.
CD34. Are there any other functions that this cell has that you guys have figured out that could maybe be at odds or complement what you're doing?
Well, it's really interesting. The CD30, if you're just walking down the street and say to the man on the street or the woman on the street, hey, do you know about these CD34 cells? If anybody answers you, they're probably going to say, oh yeah, that's the hematopoietic stem cell, which it is. CD34 cells, when you get a stem cell transplant, if you've had your bone marrow wiped out with chemotherapy or radiation for cancer, and you get a stem cell transplant to reconstitute hematopoiesis, it's the CD34 cell that does that. So it is a true stem cell. That is that it can do multiple things, if you will, when it grows up. One of the things that the CD34 cell can do in certain contexts is reconstitute all the elements of the blood. But another function that it has is to build the tubes, if you will, that carry the blood. And this is, pretty typical in embryology, that parts of the organism that reside next to each other or are related in some way often come from a common precursor. And so this is an example of that, where the blood and the tubes that carry the blood derive from a common stem cell, and that's the CD34 cell. We don't, some people have asked, do these cells reconstitute other things? Like, might they make heart muscle cells when you put them in the heart, or might they make skeletal muscle cells when you put them into the skeletal muscle of the leg? I would like for that to be true, and we tried to find evidence that was the case, but really we could never convince ourselves that they did anything more than building blood vessels. So at least that's as far as we know right now.
How about for cancer? I know that cancer microenvironment can be hypoxic, and this could cause cancer to induce angiogenesis on its own.
Blossom. Yeah, sure. Yeah. Look, I think that's a very important question. I started out life, when I was doing therapeutic development, earlier on, it was gene therapy. And people were very worried about using gene therapy to stimulate angiogenesis. And here you have companies developing VEGF, vascular endothelial growth factor blocking drugs to treat cancer. And here we are, a bunch of cardiologists figuring out ways to try to increase VEGF expression in cardiovascular disease. And it was, the question about, are you going to progress cancer, cause cancer, or, eye disease, retinopathy, proliferative retinopathy. And, over the years, we've been very, very studious and careful about, monitoring patients, excluding patients who we think are at risk, et cetera, et cetera. We've never seen any increase in cancer. In fact, before we started doing this stuff in patients, we asked the question in some animal models. We would put a cancer into an animal and then give them VEGF or CD34 cells and see if the cancer got worse. And even the animal models, we couldn't see any evidence of that. That doesn't mean it couldn't happen, but at least we were never able to generate any evidence that it was happening. We still watch very carefully. And remembering that we're treating patients who are really at the end of the road, the sickest of the sick. And so, we're trying to keep an eye on the balance of risk and benefit, even though, as I said, we've not seen any increased risk of cancer in these patients. We'll still be very careful with our surveillance. You talk to the cancer docs, and some of them have done some of these experiments too. And I think, as much as angiogenesis is a factor in some cancers in helping them to grow and spread and so forth, cancer is, much more sophisticated disease than just blood vessel growth, right? There's other features that have to be present in order for a cancer to really blossom. And so, I think fortuitously for us, we've not seen any evidence, but it's something we're always going to keep our eye on. There'll be a lot of post-marketing surveillance to see if that's the case. Because, if we're going to start carrying this, further back into patients in earlier stages of disease, we're going to have to make sure that we're not causing any downstream problems. In these patients with end-stage disease, about to have a leg amputated or crippled with chest pain, I think people are going to be more willing to take a risk of some long-term consequences in that setting. If you start talking about really more preventative type strategies, then your risk tolerance is going to be lower. And so we're just going to have to keep monitoring and collecting data to make sure that we're not doing anything that has a long-term implication that's negative.
Yeah, actually, I was just about to ask you about this. I'm sure there's going to be pressure from people that are less sick and eventually all the way up the scale to, again, people that are healthier, athletes that want an edge. But even people that are less sick, that are not at the very end stages, they're going to want this help. So how do you respond to that need? How is this going to follow some kind of path or morph in time to be used for more and more people at more stages?
Only if we, look, I'm obviously very interested in exploring ways that these cells could be used therapeutically, and hopefully that means even at earlier stages so people don't get into hot water like critical limb ischemia or heart failure. But the only way that we're going to be able to advance those types of ideas is through clinical trials. That's all. We've been very rigorous about performing these studies in a blinded, randomized way, so that when we see something, we know that it's valid. And we're excited about exploring other indications, but it's going to have to be in the context of doing careful clinical studies.
Yeah. Have you seen anything else that people can do to encourage this to happen, at least somewhat on their own, to fix ischemias? Any diet or exercise correlations or other things they can do?
I think that so exercise does increase your CD34 counts in your circulation. That's been shown. And there are some dietary maneuvers, but I would say that it's not at the point where I would prescribe it, right? So people have made observations about certain foods that may be associated with increasing CD34 counts. That doesn't mean that you're going to have better vascular repair. So I think it's something that we'll look at, but I think there's really nothing more than very casual observational data at this point to go by, which isn't much.
And do we understand the signaling that goes on in an area that is becoming ischemic to signal the CD34 to be released and brought to that site?
Yeah, so the beacon, if you will, for CD34 cells seems to be SDF1 or stromal-derived factor 1. And the receptor on the CD34 cell is called CXCR4. It's chemokine receptor. And so SDF1 expression is increased in ischemic tissue, and so the CD34 cells expressing CXCR4 home into that tissue, and it seems to tether them there. That's what seems to get those cells to hang around in ischemic tissue. If you will, the magic of how the CD34 cells do what they do is still being understood. I think the beauty of cells, two things. One is that they have an array of mechanisms at their disposal. So they're not limited. Back, like I said, I started out doing gene therapy, and we would pick one gene and overexpress it and see if that had a therapeutic effect. The cells aren't limited in that way. They can make a lot of stuff. And the more we study, the more we learn that they're capable of doing. VEGF is but one of the things that these cells secrete. They secrete exosomes, which are little like photon torpedoes of information, including microRNAs and RNA and proteins and so forth. The CD34 cell exosomes seem to play a role in their ability to stimulate new blood vessel formation. So we're still, really trying to unravel all the mechanisms. Does that mean that we could figure out how to make sort of an artificial concoction that imitates CD34 cell biology? Maybe. Although, the thing that's really hard to model is that once the cell is in the tissue, it's interacting with the tissue environment, as you were saying before, talking about cancer. And so it can change its repertoire of things that it's secreting or doing in a way that I think would be virtually impossible to decipher. Certainly you're never going to be able to figure that out in a patient, and whether or not figuring it out in an animal is relevant, I think is somewhat questionable. But but we are gradually peeling back the onion, figuring out what it is that these cells do that makes them so good at improving blood flow.
Okay, well, very good. What's I know I'll question you to death, but so what's the best way for folks to learn more about these ongoing trials? And I don't know if there's any room for participation, but to find out more about Caladrius, how can they do it?
Yeah, so that's a very, very good question. So we've been, building things on our website and on our social media accounts to inform people about the things that we're doing. And we're working on some other strategies to really get the word out from an educational standpoint, just so people understand, what are we doing, why are we doing it, what's the science, what's the evidence, and so forth. So there is some information on our website now. People can read about our clinical trials on clinicaltrials.gov, and I think there's a lot more to come in the next few months. There'll be much more information out there.
Very good. Well, I appreciate you coming on the podcast, and
oh, my pleasure.
It's great.
My pleasure. Absolutely.
Yeah, great work you guys are doing.
Great. Yeah, thank you. Great conversation. So I appreciate it.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.